The human brain is the most complex part of the body, composed of about 86 billion neurons, which is about the same number of stars in the Milky Way. Each neuron uses tiny electrical impulses and chemical signals to transmit information from between different areas in the brain and nervous system. Now, what if a neuron were able to work on command? What if you were able to control what a neuron did and when? This isn't science fiction. Scientists have been able to create a special synthetic biological technique called optogenetics, which is used to activate specific neurons or single-celled creatures. It is also used to control the activity of neurons and other cell types using light. Firstly, how does a neuron transmit signals? Certain proteins on a cell's surface are called ion channel receptors, which are like switches that start and stop electrical signals traveling down the cells. They're activated when molecules like neurotransmitters bind to the receptors, which allows charged atoms or ions to move into the cells and generate an electrical current. This allows signals to be transmitted. Scientists have been able to control how to open ion channels for years, as this would allow changes in neuronal function, allowing for potential treatments of neurological conditions. During the 1970s, scientists Richard Henderson and Nigel Unwin discovered that a bacterium produced a type of opsin called bacteriorhodopsin, which opened its ion channels in response to green light. For example, algae are single-celled organisms that have evolved to swim towards light. When blue light is shone on the eye spot of the cell, which opens a channel, sending electrical signals to propel it towards sunlight. If this sensitive part of the algae were cloned and then added into the neurons using genetic modification, those neurons could be made light sensitive as well, changing the behavior of the animal. Scientists have been able to test this on mice and lab rats. After genetically modifying the neurons in the mouse's brain, fiber optic wires are fed through a hole in the skull and used to transmit micro-precise light pulses, which last just 0.0001 second, directly to a specific group of cells. This triggers the ion channels to open, sending signals. Patients who suffer from anxiety have abnormal communication between two parts of the amygdala, which is a cluster of cells located at the base of the brain. But it is difficult to determine whether this is the cause or effect of the disease. It is possible for optogenetics to be used to aim for the same pathway in a mouse and observe the results. It was confirmed that finding specific neural circuits can trigger dramatic changes in the animal's behavior. Currently, scientists are working to use this technology and apply it to humans to treat neurological diseases. Optogenetics has truly shed some light on how we can make the world a better place for humanity.